Hello, everyone. Oops, I'm making a mess here. So welcome to the, I believe this is a Network Science Center seminar, right? Or how do we have a name for it, or just that's it? But we have to say network science, right? Because we're very proud of having a Network Science Center at CEU. And so my name is Laszlo Barabashi, and it's a pleasure to see all of you here. And it's a real pleasure, actually, to introduce Reka Albert. I don't get the chance to do that too often, right? Not lately, not in the last 15 years. And that's because Reka is from Transylvania, and as some of you know, actually, she was probably the most successful student I ever had from her generation, right? From the later generations, we have other successful students. So Reka got her, did her undergraduate degree in Kolozsvár, and then she came to University of Notre Dame, where she did her PhD, and we had together a fantastic run, and it was really kind of really beautiful years when we could actually work first in granular media, and then we saw the light and started working in networks. And I remember that when I asked her to kind of join me in this network endeavor, I kind of said, you know, I don't remember whether there's any good high-impact paper will come out of it, because I couldn't get my papers, but you know, it's a fun subject, so it might as well go ahead and do that. And she graciously agreed to join me in a project after having a Nature and a PRL on one that had no prospect whatsoever to land a good paper. And the rest is history. So what's probably often confusing for those of you who were not in network science, that she is the Albert from the Barabashi-Albert model. And I always have to point out when I speak about that, and after Notre Dame, well, at Notre Dame, first of all, I should say that in addition to the kind of media, she and I worked together virtually on all of them on the first 5 papers that have contributed greatly to development of network science, the development of the model, the discovery of the power law, then on the metabolic networks, you were in that. And then, of course, she wrote this very high impact review of modern physics paper, and that was kind of the first overview of the field. And it's fair to say that even today, all books and kind of reviews are following the structure that she has established in that paper. So in a way, the canon was born there out of that work. After Notre Dame, she went to Minnesota, right, to do a postdoc on biological physics. And that's a topic that stayed with her since then. Then she ended up from Minnesota going to the Pennsylvania State University, where she very fastly grows in the ranks, and she's professor. She may have some other fancy titles that I don't know, but yeah, distinguished professor and so on, that is very highly deserved. I mean, I think it's fair to say that it's very hard to find in her generation a woman student, a woman scientist that is as accomplished as she is and had as much impact in science in general as she did. At Penn State, she actually focused very much her work on biological networks, and she pioneered an area that turned out to be increasingly important for us, of how you use Boolean networks and network ideas to match it very closely with the experimental work, and really how you make experimentally predictable prediction using network tools. And this is crucial, because network science is often, let's say 99% is an observational science. So we describe the phenomena, but has been often difficult for us to make predictions that can be experimentally tested. And her work may have been the first in that space, where she used the tools of networks and made predictions that the experimentalist could actually test and get, do the full circle that we require for true evidence. So in that respect, her work plays a really important role in the pyramid of network science, in kind of getting direct experimental validation for the ideas that we all have about networks. And she got quite a number of awards. I don't remember all of them, but I remember a very significant one. Recently, she was elected in the Hungarian Academy of Sciences. And she's now actually spending a sabbatical in Budapest, so she's going to be here. So if you want to talk more beyond this, I think tomorrow she's going to be here and stuff like that. So there are opportunities for you to interact with her. And without further ado, it's a real pleasure to have you here. Thank you.
Thank you very much, Laszlo, and thank you for having me. It's really great to be here. So as Laszlo said, I have a group whose constituents are coming from all different backgrounds, physicists, yes, but also mathematicians, biologists of all different kinds. And what we are doing is we are doing predictive modeling. We are collaborating with experimentalists to develop predictive models of biological systems, and our goal is to have very concrete predictions that can be tested and are tested experimentally, and then those predictions are validated or you know, they can either validate or invalidate the experiments can either validate or invalidate the predictions, and that gets to iterating the model, making the model better. So while we are doing this very concrete predictive modeling, we are also looking at general insights. And in this talk, I will focus on the general insights. I will not give a lot of biological specifics. But I do want to share with you a couple of general ideas that got inspired from this specific work. So I want to start with showing you a movie which may have some difficulties. So I want to prepare you what you will see. So this is a unicellular organism called dicty. It's a eukaryote, so it's a slightly, it's unicellular, but it's of the more organized kind. And there is a micropipette which contains food for this organism. And where you see that light shadow, that's where the micropipette is, and that's where the food will be. And this organism will follow the food. So what you can see is what they do is, so the cell will start moving towards the food, and then they move the food along like a big distance apart, and then the cell will start moving in that way. But it's not moving like an automobile or like an object. It's not just changing directions and going that way. It's growing in that way. So it's reorienting itself, like it's making a new head, and then it's making new legs, and it's kind of growing. And like it's almost like a fluid, but it's not like a fluid because it's much more organized. So it's somewhere in between what you would call an object moving and what you would call a droplet of fluid, kind of just oozing that way. So I would say that even from this movie, we can see, even if we don't have a lot of prior experience with cells moving, is that cells receive information from their environment. In here, the cell received the information that there is food somewhere in a direction. It's not very close by. And this the cell is doing by receptor proteins and the external molecules, the molecules from the environment binding to these receptor proteins. They can also receive information from their internal state. And then the cell makes decisions based on this. Here, the cell made the decision to reorient itself and start moving in another direction. They can change their behavior following this decision. So how do they do this? How do single cells do this? Of course, they don't have a brain. So it helps to understand how they do this. It helps to see this drawing, which is a scientist's drawing. It's close to reality, and it shows you how crowded the internal environment of a cell is. So what you see here, these green things are the receptor proteins that I mentioned. So they are in the membrane of the cell. By the way, this drawing is not of the same cell. It's a slightly less complicated cell, but it just gets more complicated. The nucleus is here. It's this yellow, and this is where the DNA is coiled up. And then where you see the white, those are messenger RNAs made from the DNA. They will code for proteins. And the purples and the blues are proteins, different kinds of proteins with different roles. So the decisions that the cell is making, they are not decisions of an individual. They are collective decisions. They are decisions of the totality of the proteins and constituents of the cell. So, in other words, cells are complex systems. Their behavior is an emergent property. And of course, this is not only true for cells, it's true for all levels of biological organizations, more and even more true. So cellular behaviors arise from the interactions between different elements inside the cell, molecules, simply said, or macromolecules. And then interactions among cells give rise to organ behavior, the development of organs. The immune response is an example of this kind of collective behavior of cells. And then interactions among individuals leads to collective phenomena, leads to communities, and so on. And the same is true. The interactions between species leads to ecosystems and ecosystem-level communities. So in all of these cases, the higher level property, which I can call function or behavior or phenotype, whichever is more familiar for you. I think I will stick with behavior because that's probably the simplest. In all of these cases, this macro level behavior arises from the interactions at the micro level. So it's a collective behavior. And it arises from the totality of the elements and interactions. We cannot say it's because of this element or because of that interaction. So for that reason, we say it arises from all of them. But does it really arise from all of them at the same level? Do all have the same amount of say? Well, probably not. And we, in fact, know that the mapping from the totality of internal and external variables to the behaviors and decisions cells make, and also at different levels, is canalizing, meaning that not everything counts the same way. And in order to give you an example of this canalizing behavior, let's think of a behavior that we would have. For example, we would walk on the street and we would look for an address. So we receive lots of information. All of our senses are in play. We see, we hear, we smell. But we are focused on one task, and that's to find that address. So we are looking at the street names and the numbers. And so our brain filters out the majority of the information. We only keep that information. Yes, I'm on the right street. No, I'm not on the right street. I should turn here. I should cross the street. So when we are focused on something, our brain filters out the majority of information. That's true at all different levels. So all of these different crossings of the scale have a large amount of canalization in them. So not really everything counts, not really every internal and external variable counts, but we cannot really tell beforehand which one counts. And that's where modeling comes in. So that's the question that I wish to answer. So how do we explain emergent properties? So there is this canalization. There must be some elements and some features of the network. Well, I shouldn't say network at this point. There must be some features of the elements and of the interactions that count the most. How do we find these? How do we answer this question? Well, we can use a network framework. In this framework, we will represent the elements as nodes and the interactions as edges. And, of course, these kinds of networks have been constructed. And here's an example which actually underlies, at least to some extent, the movement of the cells that you couldn't see. So in this network, the beginning is a so-called G-protein coupled receptor. So it's a certain kind of receptor, and this receives information from the external molecules, like the CMP, the cyclic AMP that the cell was receiving. And then there are lots of protein-protein interactions that converge at three kind of virtual nodes or different kinds of proteins, like F-actin and directionality and myosin. So these are different kinds of structural proteins. And the very end, you can see this, there is a virtual node called chemotaxis. That means behavior towards a chemoattractant, a behavior, movement towards food. So there is a node that represents the outcome of this whole system. And this is regularly done in so-called signal transaction networks. There is a node that represents the outcome of the whole process. And here's another example. This is also a signal transaction network, and the signal here is a growth hormone. And the response is proliferation, that is cell growth and division. And the example shown here is when this response is dysregulated, meaning that the signal actually is not present, the growth hormone is not present, its receptor is present, and it's overactive. So even without receiving the signal, it's activated, the whole pathway is activated, and in red shown here are other dysregulations and mutations that can all together or even individually lead to incorrect proliferation of cells and ultimately to tumor growth. So in both of these cases, we represent the system as a network. And then we will need to relate the system's behavior to this network. And then we can find out which features or connectivity patterns in the network are critical to the behavior. And here, critical could be both in the good sense and in the bad sense, or critical to a correct behavior, meaning that it's good, or critical to an incorrect behavior, meaning that we should target it in order to make the incorrect behavior disappear. So the same idea with a little bit more detail, we will use dynamic modeling to make this connection. So in dynamic modeling, we first need to determine and construct the relevant network. Usually we cannot construct a whole genome level network of everything that a cell does. So we pick one. We pick one behavior, and we pick one or several signals that are known to be starting points of this behavior. The network will show the totality of interactions that are possible to happen. They don't have to happen at the same time, simultaneously. The edges do need to be directed, so have directions, and they need to mean either causality or even more mechanistic detail, meaning that there should be a source and a target of that edge. Unfortunately, symmetrical edges are not as informative in this case. Then each node is characterized with a state or activity level, so an additional variable that's telling us about what it's doing. If a node state is 0, that means that it's inactive. All of its edges is as though they are missing. So nothing will happen that this node was supposed to be doing. It's not happening. This is a special state. It doesn't really matter what kind of variable we are using that, but the variable level of 0 must be present. Then each node is also characterized by a function that's telling us how the state of its regulators, its neighbors, is changing the state of the node itself or regulating the state of the node itself. Here, its own state can also be part of this function. Usually it isn't. Then the long-term states of the nodes, so the states at a long time after the start of the process. And here it doesn't have to be the state of all the nodes. It can also be a subset, and I will talk about this. So the long-term states of the nodes or a subset of the nodes can be related to the higher level behavior. So this is the connection. The states of the nodes will be the connection to the behavior. And then this model can be used to make specific predictions. For example, how would the perturbation of a node or of the regulatory function of a node change the dynamic repertoire of the system, the behaviors of the system. Here we can use this to address what would happen in a disease, for example. Here, this answer can indicate us what are the important nodes that we should be very careful of, we should defend them, or also, if the behavior is an undesired behavior, it gives us targets. What are the points where we should target a manipulation or an intervention to make that behavior disappear. And also this model and this kind of thinking can lead to general insight, answering the question which features of the network are most responsible for the behavior. So far this was the motivation. Are there any questions at this point, anything that's not clear or you don't agree with? Yes, go ahead. So the timescales vary. So if it's a signal transaction network, then probably the process is happening on the timescale of minutes. Then we would wait for half an hour or an hour. I will also introduce a more technical term of the attractor and kind of give you how these long-term behaviors live inside the model. Okay, so the dynamic model is built from experimental data, and it's tested on experimental data, both internally and after the model is finished. So let me take you through these steps. So I told you we cannot model everything at the same time. We need to choose. So we choose inputs and outputs. So the inputs are the external signals, and the outputs are the outcomes. So usually we do use those virtual nodes which represent the output itself. Then from the literature, we compile the nodes that are known to participate in this process, and this is an iterative process, and we add more and more as we read more and more. And at the same time, we also synthesize the edges. There is quite a bit of an assembly process required here, but at the end, we do have a network. And then we need to develop the regulatory functions. This is also based on the literature. So information on the interactions, regulatory effects, kinetic information, interaction information, if there is such information, comes into the construction of the regulatory functions. Then we need to have an initial state, like a resting state of the system, and state data from the literature come into this information. If not, we should probably try all the beginning states that make sense here. And then using this, starting from this initial state, we use the regulatory functions to find the next state of the system and the next state of the system and so on. And we find trajectories of the system and long-term states. And we can compare this, what the model is saying, with what we know from the experiment. So state data, again, is used to compare the model's predictions. At this point, these are more like validations, not real predictions, because this data exists. So this is how we validate the model. We compare it with what we know already. And usually there are discrepancies, and in this case, we need to go back and change some of our assumptions. And this is completely valid because usually there isn't enough information in the literature to completely pin down the network and the regulatory functions. So this kind of iterative process helps us to refine the functions. And when we are satisfied with the model, we can use it to make real predictions, predict cases that were never studied experimentally, and this is perturbations, interventions, scenarios, what-if scenarios in general. And this is where the model is giving predictions, which then can be tested by follow-up experiments. And here at this point, either everything is great, or if not so much, then you need to go back again, and you iterate this process. So that's the general idea. At this point, I didn't say anything about what kind of regulatory functions to use. Let me give you an example, which may be more familiar because it's not biology, it's humans. It's disease spreading on human contact networks. So in the network, the nodes are individuals, and the edges are the disease-specific physical contact. So whatever disease we are modeling in this case, the relevant physical contact will represent the edges of the network. And the nodes can have two states, susceptible and infected. Let's just focus on the infection process and not care so much about the recovery process. So this will definitely not be a correct model, but let's just focus on this, so for simplicity. So let's say that at the beginning, everybody is susceptible and a single individual is infected. This infection will propagate through the network because the infected individual's edges will carry the disease, and the neighbors of the infected individual will be infected, and at some later point, a fraction of the population is infected. And then we can also think about what will happen at the very long time, but let's not do that yet. So two states, susceptible and infected. Any edge of an infected individual can carry the disease, so any infected neighbor of a susceptible individual can infect this susceptible individual with a certain probability. It's not a certainty. So for example, for this node A here, which has 4 neighbors, which I denoted B1 to B4, we can describe the probability of A becoming infected, and it will become infected if any one of its neighbors is infected. So the probability of not becoming infected is the probability of all of its neighbors being susceptible and not infectious. So, and 1 minus that is the probability of it being infected. And this, in this case, is bigger than 0 because actually here one of the neighbors is infected. So sooner or later, this node A will become infected. So the data to construct such a network is, we need data about the contact network, we need data about the infection probabilities, and also the disease status in time in order to see what are the time scales of the process. But in this very, very simple case, we know one thing for certain. If this is our network, because this network is connected, because these probabilities are not zeros, at the very last time, everybody will be infected. And that's because it's a connected network. If it weren't a connected network, that would not be the case. So that was kind of the warm-up. And now let me tell you about the actual model framework that we are using, and that's Boolean models. Very similar, because there are two states in this case as well, which logically speaking are denoted as 0 and 1, and in the biological cases, there is usually a good interpretation of this. So it could be different conformations of a protein. It could be different states of a channel. A channel can be open and closed. It can be active, inactive proteins. So different conformations usually do correspond to different activity levels. Or if it's a gene, it could be transcribed or not transcribed, which sometimes it's called expressed or not expressed. If in reality, there is a continuous variable, then you can think of having a threshold, and if the level is below the threshold, that's the up state, and it's above the threshold, it's the on state. So obviously, this is a big simplification. Still, as you will see, this is still a rich modeling framework, which allows us to answer some questions. So the regulatory function, the same thing. The future state of a regulated node depends on the current state of its regulators, and it may depend on its own current state. But usually, these are not autonomous nodes. They don't have autonomous dynamics. They receive information from each other. One node's output is the other node's input. So the information will propagate. So there isn't that much memory. The regulatory functions are described by Boolean logic, and they can also be described by tables. That's the simplest case, unless it would be a very big table. So this is the case where there are two regulators, and the function is the OR function, which means that if any one of the regulators is in the on state, the one state, then the regulated node will become on in the future. In Boolean models, there is no continuous time, so time is not really an explicit variable, it's an implicit variable. Instead of time, we have updates. And I would rather not go into this, but if anyone has any specific questions at any point about the update, I will be happy to answer that. There usually is some stochasticity in the implementation of time in our models, and this is because we want to try many different possibilities. And for this reason, we also do a large ensembles of simulations in which we try different things in a different order. So starting from an initial condition, the behavior of the system or the state of the system will change, and sooner or later, it will go into a so-called attractor. There are two kinds of attractors. One is a fixed point or steady state, which is a state that doesn't change anymore. So nothing changes anymore. And the other is a complex attractor, which is a set of states. And the system will revisit these states, but it will never visit any other state except for the ones in this set. So it's either a single state or a set of states. The system goes there and it's trapped there. This is inevitable in a Boolean model because the Boolean model is a discrete model. The number of states is finite. But this is also the case in a very, very general framework, a very general class of dynamic models. These models have attractors, which means that the variables, even though they are continuous variables, they can be continuous variables, they will not increase without bounds. There will be some state in which the system goes into and stays there. So is the idea of an attractor clear? Yes, go ahead. We can have limit cycles. So the complex attractor, one type of the complex attractor is a limit cycle. So here I kind of gave this general definition. So if the system keeps revisiting the states in a cyclic manner, that's a limit cycle. But because of the stochasticity, it could also do this irregularly. So that's why the complex attractor is supposed to cover both of those cases. Yes, go ahead. Well, yes, in the beginning, yes. Every state is a valid initial condition unless the biology is telling us that what the actual initial condition is or what some parts of the actual initial condition are. But you can do this systematically. Every state is a valid initial condition. But what's the next state depends on the regulatory function. And I will immediately show you an example where I think this will kind of come to life. So here's an example, a simple system of 5 nodes. And in these nodes, in this system, node A and B are designed as inputs, so their state will not change. Whatever we start from, they will keep this state. because they don't have any regulators. So these are the signals, the stand for the external signals. And then there are 3 nodes which do have regulators. And one of the nodes is not affecting anything else. It's a so-called sync node of the system. This stands for the outcome of the system. And I put the Boolean rules next to the node. So it's an OR function for both of these nodes. So there is, in fact, this node C has an external input and also an internal input from D. The same thing for D as well. And let's specify an initial condition. This is not a real system, so we can choose freely. And let's say that node A is on, so that's what I'm showing by the blue color, and the rest are off. So can someone guess what's the next state of the system? So don't worry about updates. There is one state which is the next no matter what.
C turns on.
Yes, C turns on. What about the next after that? D, is there any other possibility? E could be also, because both have the chance, so if it's their turn, they will turn on, and whether or not it's their turn depends on the duration of the edge, or the time delay, or technical details of the model. But, so it's two possibilities, but after this, the next possibility is that both of them are on. And anything else possible from now on? No, B cannot turn on. It started off, it will stay off, and nothing will turn off. So this is, in fact, a fixed point of the system. It's one of the attractors of the system. Nothing will change from this point on. It's easy to use the model to find all the attractors of the system, and this is what I call the attractor repertoire. And this is the repertoire of the system. And on the top, I'm saying the different signal configurations. So when A is on and B is off, this is the attractor, fixed point of the system. This is the one when A is off and B is on. When both of the signals are on, there are 2 possible fixed points, and this does depend on the initial condition. These 2 attractors didn't depend on the initial condition. All the initial conditions led to the corresponding attractor. Here, this is the case where the initial condition does matter. And so in one case, the 3 internal nodes are on, and in the other case, they are off. And finally, when both signals are on, then every node is on. So this is the attractor repertoire of the system. In a real case, when it's not a made-up simple example, each attractor of the network, or maybe group of attractors which have the same property, for example, all the internal nodes have the same state, can be associated to a cellular behavior. So when this is a real example, some of these attractors will mean a cell type, and others will mean a different cell type. So there is a functional biological interpretation of these attractors. The model can identify the states and also the state trajectories that lead to each of these attractors. And this leads to an informative derived network or follow-up network, which is the state transition graph. In the state transition graph, and it's referred to this way, so it's not called state transition network, state transition graph. So in the state transition graph, the nodes are the states of the system. So here there are 5 nodes, so it's 2 to the 5th states in the system. And here the node labels, yeah, I think you can see them, are A, B, C, D, E, the states in this order. And the edges are the allowed state transitions. So allowed by the regulatory functions, the choice of the regulatory functions canalizes what is possible, what's a possible next state. Also, the choice of the timing has an impact here to some extent. But for example, it doesn't change the fixed point. That's what I'm saying to some extent. So here, I will be choosing that one node can change state at any time. So that's how these were drawn. So you can see that there are four parts to this state transition graph, and they correspond to the different combinations of the signals. And each of them, well, they mostly lead to an attractor each. So the attractors, the fixed point attractors, will be shown as states that don't have any transitions going out, only a loop, so a self loop. So that's a fixed point. There are no complex attractors in this case, but they would show up as strongly connected components in this network, in this state transition graph, with no out component. So terminal strongly connected component. So the system must get trapped and not escape from that set of states. So that's not here at this point. So something interesting happened also in this simple case. There is a configuration in which there are two attractors of the system. And here we can see what are the states that can reach only one of the attractors or both of the attractors. So this state here with the blue outline can only reach one of the attractors. And the node with the red outline can only reach the red attractor. But the rest can reach both of the attractors. And the probability, so this is a Markov system, and one can calculate the probability of reaching one attractor, and also the time it takes to reach one or the other attractor. So this state transition graph is a very informative network. Any questions? You have a question? Yes, go ahead.
So the way you achieve the Markov state is that you update asynchronously,
Yes.
or you put noise on the function?
No, it's only the update. The functions don't have noise. So here, each node has the same probability of being updated. You could also change this. You could have different probabilities, and that will have some changes to this state transition graph. But the functions are deterministic. So far, we did not have any reason to mess with the functions. We are trying our best to find the best functions, and we are keeping them. Also, it makes it just so much more complicated if the functions have noise. So this amount of noise works for us. Okay, yes. So the model can also describe what happens in case of deregulations, mutations, or interventions. For example, if D is 0, that's what I'm showing with the red outline here. There are still 4 attractors of the 5. 1 attractor disappeared, and 2 have changed. So of course, if D is kept off, then it cannot be on. So this attractor and this attractor is the correspondent, the changed correspondent of the case where every internal node was on. So the repertoire changed a little bit. So that's the idea. This was a toy example, and now we are using it for real systems. And I will just give you a little bit of one real model that we have developed, and that's modeling T-cell survival in the context of the disease LGL leukemia. So these T-cells are there to find infections, viral infections. They are very, very strong. They are killing cells. So they are killing infected cells. So they are killing their own siblings and so on, the ones that are infected. That's their job. And after the infection is over, their job now is to die. So they are not needed anymore, and they need to undergo the process called cell death or apoptosis. So there is a process called activation-induced cell death, which these cells are supposed to follow, but in this disease, they don't. These cells survive. And after a while, they start attacking healthy cells, and that causes the symptoms of the disease. So we wanted to understand why the system goes awry, why isn't the program followed. And we first constructed the network that corresponds to this activation-induced cell death. And also we added to this network some nodes and interactions that we knew were happening wrong in this disease. We constructed a Boolean model of it, in which there is stochastic timing. And the model could reproduce the known outcomes, which is apoptosis, which is the normal expected outcome, and the abnormal survival state, which also has a large number of proteins in the wrong state. So there is a whole state, abnormal state, which we could describe with this model. It also reproduces some known key mediators of this process. And then we focused on seeing what is the minimum number of deregulations or changes in the initial condition compared to resting T cell, which can cause the disease, and we answered this question. And we also looked at what are the interventions that are able to make all the cells die. And we found 19 single node interventions, so a much larger number that was known at that point. And many of these predictions were validated experimentally in vitro, but in the real cells coming from patients. So this is the first step towards developing some therapy. Of course, there are many more steps. But the implication is that we have identified therapeutic targets which can be used for future therapies. There is currently no therapy, specific therapy for TLGL leukemia, only just general immune response suppressing therapies. So just to give you a little idea, this is the network. It has 60-something nodes. It's relatively complicated. There are 3 signals in the system, and we looked at 3 outcomes, but apoptosis is the most important. Then with the dynamic model, we simulated an ensemble of cells starting from different initial conditions, but still, which are still correspond to resting T cells. And then in our ensemble of models, whenever a simulation reaches apoptosis, it leaves the ensemble, and whatever we are showing is on the cells that are still alive. So for example, we could show that in the population of cells which are still alive, a protein called JAK is upregulated in all of them. This is consistent with what is known experimentally. Proliferation stops. Another protein called interleukin-2 is just transiently increased, and then it goes away, and so on. Most important is this. This is the percentage of cells that reach apoptosis. That's what they are supposed to reach. It's around 70%. So 30% of the cells, of the simulated cells, stay alive indefinitely. So it's a mixture of the 2 attractors. The majority reach the correct attractors, but a strong minority stay in the surviving state. So we developed a method to simplify the network without having an impact on the dynamic repertoire of the system, which I don't have time to talk about, but I can talk about it offline. And we could simplify it to these 6 nodes. And then we could construct the state transition graph of these 6 nodes. For the original, it's not practical. And we could find the 2 attractors. So the blue is the normal attractor, the apoptosis, and the red is the disease attractor. And find all the possible trajectories that lead to one versus the other. So here the coding is the same. The blue nodes, blue states are the ones that converge only to the normal attractor. They are the majority. Just 2 states go to the disease attractor exclusively, and the gray ones can go to both. So they could go either way. We could calculate the probabilities of going into one versus the other. So overall, the normal attractor has a much higher probability of being reached, but still there is a strong minority there. And then we could use our model to predict interventions that would ensure that all the cells, so the surviving cells, also die. And this is how it would happen. So this is the fraction of cells that undergo apoptosis. It stops in the normal situation around 70%, but when we use that intervention which we developed, then the percentage goes up to 1 or 100%. So 19 new predictions here. Any questions about this? Okay. So now I can show you another insightful derived network. And this is needed because the state transition graph, although it is very insightful, but it can be computationally taxing to construct. Now with the simplification of the network, we do have some choices, but I think this other choice is also a really good one. So we can integrate the network with the Boolean regulatory functions. And this gives us another network which we call the expanded network or the logic expanded network. So what we do for this is we first eliminate negative edges by adding complementary nodes or negated nodes. So every node will have its negation as well. And the regulatory function of the negation is the negation of the regulatory function of the original node. This way there are no more negative edges because every negative edge becomes 2 positive edges involving negated nodes. So if A inhibits B, the negation of A will activate B, and A will activate the negation of B. This is very useful because now there are no inconsistencies. Sometimes it happens, or often it happens, that there is a positive path and a negative path between the same 2 nodes, and then we don't know what to do with them. This way, this doesn't happen anymore. The other is to eliminate the ambiguity between the AND and OR gates by explicitly showing which one is the AND gate, and we do this by adding a composite node. So when there is this black dot, the composite node, it means that the gate between A and not B in this case is an AND gate. And whenever there is nothing, that's an OR gate. One problem of the matching between a network and the Boolean model was that there is no one-to-one matching. There are several Boolean functions which are consistent with the same network. Now that's not the case anymore. This expanded network is a one-to-one correspondence with the set of Boolean regulatory functions. So why is this useful? Oh yes, so before that, let me tell you that these composite nodes, these black dots, have special properties. They are not all like the original nodes, and that's because they represent AND gates, so they need all of their regulators. Loss of any of the regulators means that this composite node can never be activated. So why is this useful? Well, we actually used it for several different purposes. One is that we can find out what are the elementary signaling modes. So elementary modes of connecting a signal and a response to that signal, which are functional and which can also work independently. So in this case, there's a signal here and there is a response here. And there are two elementary signaling modes. One is a path, a simple path, but the other is a subgraph. So the blue is a subgraph. And it has to be a subgraph. The path itself would not work. So this path would not work because it involves a composite node. The composite node needs all of its regulators to be present at the same time. So it would not work with just one of its regulators. And for that reason, this whole thing, this whole subgraph, is an elementary signaling mode. Nothing less than that would be able to carry the signal and generate a response. We can also find out how many elementary signaling modes a network has and how many of them are independent from each other. And to our surprise, we found that for most signal transaction networks that we constructed, there is only one node independent elementary signaling mode. So when we put down one, there isn't another one that's independent, meaning that in these cases, it's possible that the disruption of a single node will disrupt all the paths between the signal and the response. And that's because of the dependency, because of the AND gates. Another insight that we can gain from this is to find the centers of stability of the network. And these centers of stability determine the system's attractor repertoire, and the information on these centers of stability can be used to drive the system into behaviors that we like or away from behaviors that we don't like. So I want to spend some time on this, because I'm very excited about it. So in this expanded network, we can find stable motifs. These stable motifs are strongly connected components, so feedback loops in general and nested feedback loops, in the expanded network. But not every feedback loop is a stable motif. A stable motif does not contain both a node and its negation, which simply speaking means that it's a positive feedback loop. It's not a negative feedback loop. It cannot involve it's not negative. None of the loops in it can be negative. And if it contains composite nodes, it also needs to contain these nodes' inputs, meaning that this stable motif cannot have AND gates in its borders. It can have the AND gates only inside. What this means functionally is that this stable motif can maintain a state. There is an associated state, which if the stable motif reaches, it will stay there no matter what the rest of the network is doing. So this stable motif is not independent of the network, but functionally independent, becomes functionally independent from the rest of the network when it reaches the corresponding state. So that's because the dependence is the AND gates, and the AND gates are only inside. So let me show you in the same simple example. And here there are two signals, so let's give a state to these signals. And the most interesting case was the case in which both signals are 0. So in this case, this is the expanded network. So every node will get its negation, and it's actually the repetition of the same between the nodes and their negations. So this is not the best example. It's maybe a too simple example, but it is illustrative, and it continues the same example. So this is the expanded network. And then can someone tell me a stable motif in this? So the C and D positive feedback loop, and also the positive feedback loop between the negation of C and negation of D, they are both stable motifs because they are strongly connected component, and they don't have nodes and their negations, and they don't contain composite nodes. So this is a stable motif, and then we can just look at this and read off the corresponding state, because if it's the node that's in the stable motif, that corresponds to the on state, and if it's the complement, then it corresponds to the off state. So this is the state C equals 1, D equals 1, and then obviously E will equal 1 as well. And this other one is the case where all three of them are 0. So we can just read off the state. We found the attractor repertoire of this system with much less work than calculating the state transition graph and reading off the sync nodes in this case. So that's the idea. This is an alternative way, which, although in this case it's not very clear, so one has 6 nodes and the other has 8 nodes, but for a bigger system, it's obviously, it has much, much fewer. So the expanded network has much fewer nodes than the state transition graph, because the state transition graph is 2 to the number of the nodes. And this is twice the number of nodes plus the composite nodes, so 3 times the number of nodes at max. So we can use the knowledge of the stable motifs to iteratively simplify the network and finally find all its attractors. Let me show you a more complex example of 5 nodes with lots of gates. So these are the Boolean functions here. And now, for simplicity, I will not show the expanded network. I will show you the stable motifs in their original form, the way they appear in the original network. So A equals 1 and B equals 1 form a stable motif. There's a positive feedback loop between them, and there also is a self loop on A. A equals 0 alone, because of its self loop, can be a stable motif. And then there is equals 1, a stable motif, and a stable motif of 3 nodes, a positive feedback of 3 nodes, another stable motif. So we can then choose any one of these and see what are the consequences of that stable motif stabilizing. And in what we do in this case is we plug in the corresponding values, and we will find that the network now only has 2 nodes. When we plug in this node, the network is still 4 nodes, and so on. So we can follow any one of these branches. In the remaining simpler networks, we can again find the stable motifs and plug in, and so on. And at the end, we will find the attractors of the system. So we can find the attractors of the system by repetitively finding stable motifs, finding the corresponding state, and plugging in the corresponding state. We can also use this information to control the system, to drive the system. Because every attractor corresponds to a succession of stable motifs. So here I'm not showing the simplified network anymore. I'm showing if this stable motif stabilizes, what are the possible stable motifs? If they're stabilizing, what are the possible stable motifs, and so on. So these are, we call these stable motif succession diagrams. They are what's the next, and what kind of branchings are possible. So in this case, there are 3 trajectories that lead to the same attractor. And they all involve the same 3 stable motifs. A equals 0, D equals 1, E equals 1. A equals 0, E equals 1, D equals 1. It's the same. E equals 1, A equals 0, D equals 1. So if we put A into the off state and D and E into the on state, we can be sure that the network stabilizes in this attractor. But there is more. We don't need to put all 3 of them. If there is a trajectory where there are no branchings, there are no choices between different stable motifs, then the first stable motifs, like a domino effect, leads to the stabilization of all the rest. So with this reasoning, we can find that D is always at the end. So after D, there are, no, sorry. No, that's not the case. After D, there can be choices, but after E, there are no choices, and after A, there are also no choices. So it's enough to control A and E. D doesn't need independent control. So we don't need to control all the nodes of all the stable motifs in a succession to make the system go into an attractor. Usually a subset of the motifs and definitely a subset of the nodes of the motifs is enough to make the system follow one of these trajectories and go into an attractor. And here's the real example of the TLGL network. So remember that there are two attractors, apoptosis, and the leukemic survival state is the other attractor. And this is the stable motif succession diagram. The number of stable motifs is not very high, and you can see that they repeat. So there are lots of repetitions in this succession. I would say there are 3 very important motifs and 4 and 5 total. So if we start with this stable motif stabilizing, there are no branchings. Another will stabilize, and the end is apoptosis. If we start with this red stable motif, there aren't even any more stable motifs. Everything stabilizes, and the outcome is leukemia. So it's very simple in this case. If we start with this node stabilizing, then there are choices. So if we want to control the system, and if we want the system to go into the leukemic state, all we need to do is to make sure that this stable motif goes into this state. Here, gray means off, and black means on. And control of a single motif, so for example, control of a single node, for example, S1P in the on state will stabilize the motif, which will stabilize the attractor of the network. So one node intervention can make the system go into the leukemic attractor, which of course is not what we want. But making this stable motif stabilize will go into apoptosis, and that's another single node intervention, S1P equals 0, for example. Or there are combinatorial interventions as well. So for example, make the system follow this, this diagonal trajectory. In this case, we do need to control Tbat. One node of this motif, it can be almost any one, and then one node of this motif. So a 3-node intervention can also lead to apoptosis. So we can use this information of the relationships between the stable motifs to control the outcome of the system. I also want to say that this is not just something that we construct. This also reflects the autonomous trajectories of the system, because the initial conditions where the system starts from and also the timing will determine which stable motif stabilizes first. And then from there, maybe there are no more choices, maybe there are some choices, then again, timing, stochasticity, different inputs could make the system choose one of the trajectories and so on. So this is also showing us the choices the system has and the choices that the system doesn't have. So I would say that this is also an informative, but not yet network. This is succession. One is coming after the other. It's not really relationships. So I can tell you about something that we are developing that's more of a network. Okay. So I will just very quickly mention another model that we developed and predictions that we made with this. And I will not go into as many details as I prepared. So this is another signal transaction network that corresponds to a cell fate change called epithelial to mesenchymal transition. And this transition happens normally in 2 important contexts. One is embryonic development, and the other is wound healing. And what happens in these cases is that cells that are initially epithelial, like skin cells, for example, which are bound to each other, they form like a 2-dimensional sheet. They cannot move. These cells become mesenchymal, meaning that they change their shape, they are not bound anymore, and they can move. So it's a quite a dramatic transition, and this is needed in many contexts, but unfortunately very bad in the context of cancer, because it's the first step towards metastasis of cancer. So we constructed the signal transaction network that leads to the loss of one of the proteins that's a binding factor between these cells. Ecothyrin is the name. So the outcome is ecothyrin, which is thought to be the hallmark of this transition. So the transcriptional downregulation of ecothyrin is the hallmark of the transition. And there are many different signals that can lead to this, and there is a complicated network. We constructed a Boolean model in which we have 2 time scales. We have a slower and a faster time scale, but that's somewhat of a detail. So the model can reproduce the known epithelial and mesenchymal states, the known markers and key mediators of the transition. And we use the model to try to predict interventions that would block the transition. So this is the network. The blue nodes here are external signals, but some of them have edges coming into them, and that's because the cell can make them as well and put them out in the environment. So the cell can contribute to its own environmental signals. The green ones are transcription factors, and these green transcription factors are downregulated ecotherine, which is here. So we found 8 stable motifs in this network. I'm showing 4 of them. Any one of them can lead to the transition. So if any one of these groups of nodes stabilizes, the network will go into the mesenchymal state. It doesn't matter what is the state of the network right now. And also, it's very easy to lead to the stabilization of any one of these motifs. So any one of the 12 signals, or any one of 27 internal nodes out of 56, so almost half of the internal nodes, can lead to the stabilization of one of these motifs, which then leads to the mesenchymal state. So this is a very, very, very robust outcome. So what it means is that it's very difficult to reverse it. We can block it with combinatorial interventions, and the simplest combinatorial intervention is to knock out 2 nodes. One needs to be SMAD, and the other can be, there is some choice in what the other can be. And here we made a number of very specific predictions, which then were validated experimentally by new experiments. So this is a real prediction. We also predicted what would it take to make the system go into the epithelial state from any initial condition. The stable motif corresponding to that state is the whole strongly connected component of the network, which is more than 2/3 of the network. But we don't need to control all of them. We need to control 5 nodes, 1 in each of these yellow domains. So control of 5 nodes, for example, knockout of 4 and constitutive activity of 1 of them, can lead to the reversal of the transition from any state, including the mesenchymal state. So that's a prediction so far. It's very difficult to do experimentally a 5-node intervention. So we found the centers of stability in this network, and we predicted combinatorial interventions to prevent the transition and also to reverse the transition. Okay. Yeah, and I think I'm very close to being done. So I think this expanded network is very useful. It doesn't really have any cons, but there are definitely some places of improvement, and we are working on that. So one is that so far we are using the Boolean framework. But actually the concept of the stable motif is not only is not wedded to the Boolean framework, and we have actually developed the correspondent of the stable motifs and of the expanded network for discrete models with more than 2 levels. The idea is very simple. Instead of having 2 nodes for every original node for the 2 states, let's have as many nodes as many states it has, so as many virtual nodes. Those virtual nodes will be described by Boolean rules. So from then on, we can use the whole machinery, and we can find the stable motifs. So the idea is simple. It seems the details all work out. So the only thing is, the more states there are, the bigger this network will be, so the higher the computational complexity. The other is, we are developing a 3rd derived network, and that's a logic network of the external signals, stable motifs, and outcomes. And this is the logic network of the EMT network. This was easy to do because it's so robust outcome. So it turns out that any of the signals can drive any of the stable motifs, and any of the stable motifs can drive the outcome. So all of these edges here mean logic sufficiency. So one is sufficient for the other. So everything is sufficient for everything in this case. We expect that there will be more ambiguous cases, and we are using other logic. We are developing the formalism to have logic of the type necessary, and also necessary inhibitor and sufficient inhibitor. So we are working on that. So in conclusion, I think this proposal to represent the system as a network and then connect the outcome of the system to the attractors in the network, I think it's working. And from what we have learned is we can say that the behavior of the system can be represented by the state of the outcome nodes and also the state of certain stable motifs. So sometimes just the outcome node alone is not able to completely characterize the behavior of the system or unambiguously characterize it. And the external signals and the stable motifs determine the behavior. So when I told you that this mapping between the external and internal inputs and the behavior of the system is canalizing, it's the stable motifs, we believe is the stable motifs that are really driving the behaviors. And of course also the external signals, so we cannot disregard the external signals. We cannot really say what subset of the external signals are important, but of the internal nodes, we can say that it's the ones that are in stable motifs that are the most important. So the stable motifs of the molecular interaction network are critical determinants of cellular behavior. So this is really true, and we only tried it in the Boolean framework, but the stable motifs mean self-sustaining positive circuits. So we believe that the general conclusion is that positive feedback loops and positive feedback circuits are important potential functional drivers in all dynamic contexts. And I'm saying potential because our experience with stable motifs is telling us that it's not all positive feedbacks are functional and important. It's certain part of the positive feedbacks that are functional and important. And that's why it's important to further develop to other dynamic frameworks the idea and concept of the stable motif. Of course, this is not really new. The importance of positive feedbacks was said in other contexts as well. Here we are saying that it's the functional positive feedbacks, and we are giving a method to find which ones are functional and drivers. So let me finish by acknowledging my many students and collaborators who really contributed to the work that I told you about. So Ren and Zhang did the TLGL model, so constructed the model, and did most of the experiments also to validate the model. Steve Steinway is an MD PhD student who is getting his MD degree in just a couple of days. He already has gotten his PhD. He constructed the EMT model and did all the experiments to validate the model. Reisheng Wang was a postdoc who first constructed the expanded network based on some original ideas by me, and she worked on the elementary signaling modes, which I didn't have time to talk about. Jorge Zanudo really did a lot in this. He ran with the extended network idea and came up with the idea of stable motifs, and he's doing the control with them. Asiya Sadatpour, she's a mathematician. She worked on the network reduction, which I didn't have time to talk about, and also the analysis of the state transition graph. Xiao Gan is now working on the multi-level stable motifs, and Parul Maheshwari, both of them current graduate students of mine, are working on the logic backbone. And thank you for your attention.
Thank you, Reka. Any questions? Let me start it up. Actually, I missed one of the points, and you may have said it. So, is finding the stable motifs computationally efficient? Or so how would it scale with the system?
Yes, so it's not polynomial. And the issue is finding cycles, because you need to first find cycles, and then you assemble the cycles to make the stable motifs. So to find those ones which have these two properties, that they don't contain nodes and complementary nodes and so on. So the cycle finding has some pretty efficient algorithm, but it's NP. So
it's NP. So
yes.
So at the end, so it kind of simplifies this with just a couple of factors, the problem,
yes.
but not actually,
yeah, it
doesn't change the scaling of the problem.
Well, qualitatively, it doesn't change the scaling. It's still not polynomial. But in practice, it's much better. So we can handle nodes or networks with 100 nodes easily. So it's not thousands.
Have you moved any of these to therapeutics? I mean, particularly for leukemia, you're making specific predictions how to kill the leukemia cells.
Yeah, so no therapies yet, because it's a very long way.
But have you started on that path, or do you have to get small molecules?
So we are still on the so-called preclinical level of in vitro and then going to in vivo. So in the TLGL, the problem is that there is no in vivo animal model, so we need to do this on patient samples, so we are still in the in vitro level. with the LGL leukemia, no, the EMT, we are on our way to in vivo animal models. Yes.
And that would be a single drug or multiple drugs?
It would need to be multiple for EMT. We actually found, so it looks like there is a single one, and that would be not targeting the transcription factors, which are just one step away, but we found that those leads to intermediary states. So because the stable motifs don't turn, it's not a good solution. It would lead to a hybrid. So there are no single node, so it would need to be multiple drugs. But two, it seems that two would do. Yeah. Yeah.
Any other questions? Yeah, please.
Yes.
Is this problem somehow related to control?
Yes. Yes, so stable motif control, we are proposing it as a method of control. And among the methods of control that currently, like structure-based method of control, there are some similarities between feedback vertex-based control by Atsushi Mochizuki, because that's also based on controlling cycles. So there are similarities between stable motif control and feedback vertex control. We like stable motifs better because it says that you don't need to control all the cycles. It tells you which cycles you need to control. And it can do that because we also have the Boolean regulatory functions. And feedback vertex set-based control wants to work on a very general framework of essentially the ensemble of all the dynamic models of a certain type.
Control is not trivial in this one because this is a highly nonlinear system, right? Yeah. So that's right. It's very difficult to do that, but there are some tools for doing that.
Yeah.
Any other questions? Please.
I would like to ask about the logical support. So most of the presentation is dedicated to first order logic, and I'm using logic in a completely different approach, and I'm just wondering, you have to explore a huge search space,
Mm-hmm.
but maybe so logicians develop theorem rulers where, I don't know, some input is a kind of set of assumptions, and
Mm-hmm.
hypothesis or metastasis are
Mm-hmm.
theorem. So it might be the case that, but I don't know whether you could try or work in this direction to use that kind of theorem ruler software to apply, because you obviously computationally use deductions, but it's a completely different philosophy, and I cannot see index what's going on here, but if it's a different approach, then maybe it tackles this computational complexity from a completely different angle. So it might be, and I have a second question. One of your last remarks is necessity and whatever, it seems to me you are moving to a model logic, because these are model logic operators.
Yes.
So
Yes.
want to go, so we have some logicians who have more.
I'm searching for logicians who would help with this, because so far this is just our work, and I've been, yeah. Oh, so that would be great. I'd be very grateful for any advice, because I can see that it's very, very necessary. I can also see that the biological papers use this language all the time. They say this node is sufficient, this node is required, but there is no framework, there is no theory behind it. So I think this theory is very necessary. I think it's doable, and if I could get some help, I think it would go much faster.
No more? Let me take
Yeah,
a
yeah.
final question there.
So I'm a biologist, maybe I don't know what's relevant for everyone. It's all right, thank you. Is it, maybe we can take it after if there's no interest for the audience, but how do you define, I'm not clear on what is active node in a biological sense? Is it concentration of a certain?
It can be the concentration, yes. So if it's the concentration, then what we are saying is that there is an internal threshold. And if the concentration is higher than that, then the node is able to do what it's supposed to do. It's able to do its regulatory role. If it's lower, then it's not able. So those are the two states. But the other is in many cases, many transcription factors are regulated post-translationally with post-translational modifications. And then there are different conformations or different phosphorylation states. And usually there are two of them, or one can categorize them with the active and the inactive conformation. This is also language that's often used. So yeah, so those are the other cases are when it's a channel or a gate, it can be open or closed. So in many cases there is a pretty natural interpretation.
Sorry, there was one more hand and I said the
Yeah.
final one, so it's fairness. So maybe
Okay, so if I cite correctly, there was a general assumption that the Boolean network itself is known and complete. Is that a good assumption?
So this is not an, well, if it's an it's not an assumption, it's a working hypothesis. And the reason we are making this working hypothesis is that we constructed this network ourselves and we put a lot of work, like years of work in it. So I think it's the best it can be right now, but it's you know, it's never complete.
So my
Yeah.
question is actually in two parts. The first is that a valid assumption or a good assumption? So for example, if I pick a random type of cancer, am I going to find enough data in the literature so that I can construct an accurate enough network so that I can do this sort of simulation?
Yeah, so if you really do want to do this, I would suggest that you start with review papers, and you can get a feel for what the field is thinking about the nature of knowledge. Usually, I would say there is enough to start, but you know, the more you know, the better. Also, sometimes you don't want to go into a field where there are big controversies because then you don't know what to believe. So if you get, from those review papers, you get the idea that there is at least an emerging recognition that this is a network problem, then probably there is enough to construct a network.
So that's actually the second part of my question, that if I take, for example, a random cancer, look for, construct an initial Boolean network, do this sort of simulation, and then I can get some recommendation if I control this and this happens and so on. And if it matches with what I get in experiment, then my Boolean network is accurate enough. If it doesn't match, then I might have the assumption that I'm missing something from my Boolean network. Could this be used to come up with some good ways to optimally refine my network? So figure out how many nodes am I missing? Am I missing a lot, only a few?
Yeah.
How many connections?
Yeah, very good
Where is
question.
this
Yes.
for us?
So we are not at the point where this can be automated and optimized, but yes, we do have good feelings. Like you get the feeling, so these certain kinds of discrepancies have certain kinds of solutions. Sometimes you know that the connection between known elements would work, and in other cases something new is needed. And there you can kind of prescribe what the new thing is supposed to do. So yeah, I don't have an algorithm for that, but I think an algorithm can be developed. It can be formalized, and
So for example, in these examples that you showed, did you have to refine the Boolean network to get the good network?
Yes. But usually it was more, we were putting in new edges. We were not putting in new nodes. Although in one of the examples, in a model that I didn't talk about, we knew that there are lots of gaps. So we had lots of black dots in our network. So those black dots could be, actually one black dot could be the same as another black dot, or they could be anything. So we knew that there are gaps. But we still didn't really attempt to put in new nodes. We were still trying to stay with the confines of having new edges between existing nodes.
All right, well, it's a pleasure to have you. Thanks for coming.
Thank you.